Light rays are parallel to each other for the most part. They're over 20 feet away. And so there's not much bending of the light that needs to be done. So the lens is going to be elongated. And we'll look at what's causing that elongation in just a moment. But both the cornea, because it's convex, and the lens, because it's convex, bend the light usually enough to focus the light on your fovea centralis, where you have the most, what they call visual acuity. In other words, your clearest vision. And so that's what you're trying to do, is focus on that little pinpoint at the back of your eye. And remember that everything gets inverted because of that bending of the light. By the time it hits the fovea centralis, or any other part of the retina for that matter, the image has been turned over and backwards. We talked about that too. Now, if you're looking at something up close, then you've got the light rays are going to be more divergent when it's up close, and that means that you're going to have to focus it more. And the cornea only focuses it so much, it doesn't change shape. But the lens does. It'll thicken, as you can see, it'll get thicker and rounder. And as a result, it can focus the light better to that point, but it has to, it has to refract the light more. And that's called accommodation. Accommodation is the lens thickening and shortening to focus light for near vision. So when we talk about accommodation, that's referring specifically to the focusing during near vision and anything less than 20 feet away. And what focuses the lens, or what accommodates the lens, or allows the lens to stretch back out again, is this: the ciliary muscle of your in the ciliary body. The ciliary body contains ciliary smooth muscle that is attached to these zonular fibers, protein fibers that attach to the outer edges of the lens on each side. And so when the muscle contracts, it pulls on those fibers, and that elongates the lens. So when you're looking far away, the lens is being pulled to make it more flat. When you're looking at something up close, the zonular fibers are relaxing, so the smooth muscle relaxes, allowing those zonular fibers to relax, and the lens goes back, to that rounder shape, which seems kind of opposite of what you would expect, but that's apparently how it works. So, that accommodation happens when the smooth muscle relaxes and the zonular fibers relax, and the lens becomes more round. Okay. Any questions about that? Okay. Now, and I talked about near point of vision, usually about 4 inches away for a young adult, but that, as we'll see, changes as you get older. And so we're going to look at a couple different types of refraction, what they call refraction abnormalities, meaning, bending the light is not being bent enough or is being bent too much. So that's why, you get your eyes checked, and they want to check your whether you're 20/20 vision for far away vision, but also they look at your close-up vision as well. If you're nearsighted, this is called myopia, nearsightedness. This is when close-up things are clear, but anything far away gets blurry. And so what's happening is the image is being focused in front of the retina instead of on the retina. So by the time the image gets to the retina, it's not in focus, it's blurry. So your focal point is not on the retina, it's in front of it. It's kind of like when you're focusing a microscope. When you get a microscope in focus, you're bringing that image to what we call the focal point. And if it's above or below that focal point, it's going to be blurry. You want to get it right on that focal point, and that's basically what you're doing with the lens of your eye. You're trying to get it right on that fovea centralis, and sometimes it doesn't always work that way. So let's look at a picture of that. There we go. So there's a normal eye. A normal eye has a certain thickness of the lens and a certain roundness. It's going to be a perfectly round eyeball, so that for far off vision you can you can get that thing focused right there in the fovea centralis. However, notice how this eye is longer, it's more elongated from front to back. That's one cause of myopia. Another cause is a thicker than normal lens, which focuses it too much. So either by having your focal point here So you're not hitting the focal point, you're hitting the focal point is here. This would be where it would normally be if it were a normal eye, in other words. So you're trying to focus it at where it should be, when in fact it's back here. Or if you've got a normal eye but thicker than normal lens, it's over focusing, so it's putting it in front of the retina as well. And that's why you end up being nearsighted. Now, so nearsightedness, you're able to, if you're nearsighted, you're able to focus those divergent rays more easily because they are divergent. Then you can change the lens enough, to get it in focus. For near vision, you just can't get that far vision corrected. So that requires concave as opposed to what they call convex lenses, and that's where the inside of the lens is concave, really. This is more flat, but this is more concave. And what that does, a concave lens, this is convex, and that bends it in. Concave bends it out. So basically, you're taking those rays, those parallel rays, making them more divergent, so then you can focus, and of course the doctor has to figure out exactly where that focal point is, how much correction you need, how concave it should be, yes.
So when people say that you're straining your eyes by like reading too much and it's going to cause bad eyesight, what do they mean? Would it cause like myopia?
That's a good question. It may have to do with something we're going to look at in a second. That's I don't know if it has to do with the lens itself or if it's something to do with the smooth muscle. That I've never really understood because it doesn't seem like it would matter whether you're reading or not.
Yeah.
But it may be that when you're doing nearsighted vision, that your eyes are having to work harder at it or something. Because oftentimes when people read a lot, they end up having to wear glasses, especially in college and stuff at college age. So, but I'd have to look into that because I'm not quite sure why that would create eye strain or why it would create a need for glasses, because that doesn't seem to make sense. But it does happen as you age. But when you're 20, you're not old enough to have bad lenses, so it should be okay. So I have to find out about that. Now, the other one, the hyperopic, meaning you're farsighted, you can see far away, but you can't see up close. And this is, something you would be born with, or have as a child, as opposed to what happens in adults as they get older. So what's happening is you have notice how the eyeball is shorter. So you either have too short of an eyeball from front to back, so that what happens when you're looking at something far away, it gets focused behind your eye instead of at that focal point. Or if you have a thinner than normal lens, that thinner than normal lens doesn't focus it enough, and it gets behind, it's being focused behind the eye, so to speak. It's not really, that's just saying that's the focal point. So what you have are more, you don't have focused light rays at the focal point or at the fovea centralis, and so it's going to look blurry. And if you correct for that, convex lenses instead of concave lenses, because basically you're adding a little bit more curvature to focus that light even more. So here you're trying to diverge the light so that you can get that light focused to the right spot. Here you're trying to get that light focused a bit more to that focal point on the fovea centralis. So as far as, that's what I know about this. I don't know, I'm not an eye doctor, so I don't know all the little other things that can happen, but these are the two most common types of eye problems. Any questions about that one? Now, there's also, you may go to the doctor and they may talk about an astigmatism, that you have an astigmatism. It's not astigmatism, it's an astigmatism. It's altogether one word. And this is where you might have a slight irregularity in the lens. It may not be a perfectly round lens. It might have some, waviness to it. Same thing with the cornea, could be wavy. And that can sometimes get, develop or get worse as you get older. And so parts of the image might be in focus and other parts might be out of focus. And you might not even notice it unless it's really bad, but your doctor might see it during an eye exam. and so they can adjust lenses accordingly, you know, for an astigmatism. Presbyopia, this is what happens as people get older and they start holding their book further away until they can't hold it any further away, so then they have to get reading glasses. And this is because the lens loses its elasticity. It doesn't change shape very easily, or it does so very slowly. Sometimes after a while, it'll elongate. You know, it might take a little longer to elongate. So after a while, the image might get a little clearer. But after a while, you know, it's just, you don't have arms long enough to hold it away from you. So, so people usually in their early 40s is usually when it starts happening. You might notice that suddenly the image isn't as clear as it once was, or that your near point of vision is a little further away. And that's usually corrected with either reading glasses if it's just slight, but if it gets worse, you might have to have bifocals or those gradual type of glasses where you can see close up, middle distance, and far vision. Particularly if you have some, you know, far-sighted, or if you're nearsighted and you can't see far away, you might have more correction there. But oftentimes people might be okay on the far vision, but it's that up close and middle vision. It's like initially it starts out, it's the near vision, and so you get reading glasses, and then you're watching a movie on TV and you can't read any of the words on the screen. Then you go, okay, my middle vision is now getting out of whack. That's when you start going to either bifocals or those gradual lenses. So that's what you get to look for. I used to have eagle eyes, now my eyes are terrible. So, you know, cherish it while you have it, because it doesn't last, unfortunately. I mean, some people it does, but many of us end up with those presbyopia problems. Okay, now we talked about this already. This is the picture. Any questions about the previous stuff before we go on? So the retina, this is, you know, we talked about this already. You've got, here's the blood vessels and the optic disc. This is where blood vessels enter and exit the choroid, but also where the optic disc is where the optic nerve is. And then over here is the macula and fovea centralis. That's where you're focusing the light. It's a pinpoint in the middle of that yellowish macula. And as I mentioned before too, there are no, as we'll see, there's no photoreceptors here. And I'll show you why in just a minute. But there's no photoreceptor, not there, I'm sorry, right here. There's no photoreceptors in the optic disc. It would make no sense for them not to be there. Optic disc has no photoreceptors, so therefore there's no vision. It's called the blind spot. And your book has a description of how you can see where your blind spot is. There's a little thing you can do, little exercise you can do to kind of see where that blind spot is. So let's move on here. We just talked about this. So this is just showing you that, you know, your fovea centralis is right straight back here. That's your focal point right there. Now, moving on, let's move on to a discussion of the retina itself, the inner layer of the eye. There's two layers of the retina. There's the pigmented layer, which is just on top of the choroid layer. And just like the choroid, it's pigmented. It's got melanin in it. So you've got the choroid, and on top of that, you've got the retina, and the pigmented layer of the retina has melanin in it. And that allows, again, the absorption of extra light in your eyes, so that, you know, light bouncing around in your eyes doesn't happen. So the light, some of the excess light gets reabsorbed, gets absorbed, so you don't have scattered light rays in your eye that would interfere with vision. And then on top of the pigmented layer, you have the neural layer. Neural as in nerves, neurons. The neural layer is where you have your photoreceptors, which are receptor cells that are stimulated by light. And then you have other neurons as well that are involved in transmitting that information eventually to the brain. And so here is the retina. They're showing it in yellow. It's not really yellow. It's going to have some pigment in it in the pigmented layer. There's the choroid. It sits on top of the choroid. So you got sclera, choroid, retina. And here is a picture of those layers. We're going to look at a more up-close image of that. But in this picture, you can see This is the sclera, the white of the eye, which also extends around the optic nerve to protect it. You can see blood vessels running down the middle of the optic nerve, which is cranial nerve 2, and those blood vessels are supplying, blood supply. You can see this is the retina right here. There's the choroid right there, but there's the pigmented layer of the retina, and then there's the neural layer. And notice all these lines, these are axons from the retina. They all converge here in the optic disc. This is the optic disc. You can see why there's no photoreceptors. It's just axons coming together to form the optic nerve. Now, so let's look at the neural layer. I'm going to show you this as you can read about this. The cells in the neural layer that I'm concerned about you knowing are the rods and cones. Those are the ones that are the photoreceptor cells, the bipolar cells, and the ganglion cells. Okay, those are the cells I'm concerned about you knowing. The other ones, the horizontal and amacrine cells, you can see them in the pictures. Those are actually interneurons. The retina actually is sort of an extension of the brain in that it has interneurons in it that process visual information. There's a lot of processing that goes on even before you get to the brain. We're not going to worry about those though. We're just going to focus on those other cells. So let's look at, this is the picture at the top. This is the direction that light is coming into your eye. The yellow is the direction coming into your eye. So that coming up from the lens will be down here and going up. Notice that the rods and cones, let me use this pointer here. These are the photocells. Oops, let me turn that on. The photoreceptor cells are rods and cones. They are deep in the retina. The light actually passes through all these other layers of neurons before it hits the rods and cones. So these are rods and cones. These are the cones, these are the rods. And that's actually the photocells that are the photoreceptor cells. Notice that the direction of the nerve impulses go this way, in the opposite direction. So these cells get stimulated, they in turn stimulate bipolar cells, which in turn stimulate ganglion cells, which send impulses to the optic nerve. It's the ganglion cells, axons, that you're seeing here in that optic nerve and in that yellow layer right there. So let's look at this picture a little bit more enlarged. Oops. Let me see here. Okay, this is the same picture we just looked at. The light is coming in from bottom up. So it's as if you're looking down, the light's coming up through your lens, and it's hitting, there's some retinal blood vessels here, but this is where the surface of the retina is. The light is penetrating all these layers before it actually stimulates the rods and cones. These are the rods and cones. They're named for the shape of what they call the outer segment. We'll look at that in a minute. In other words, this part is called the outer segment. It's cone-shaped. And in the rods, it's rod-shaped. So that's where the name comes from. And then, again, those cells stimulate bipolar cells right here, which are literally bipolar. Dendrite on one side, axon on the other. Those bipolar cells then stimulate ganglion cells to generate impulses to the nerve, to the optic nerve. And these axons extend all the way to the optic nerve and onto the brain. Let's see, there's something I was going to say. Oh, we're going to look at another picture that shows these layers in actual retina. So notice you have the photoreceptor layer, and by the way, the top, that's the pigmented layer. That's the pigmented layer that is in contact with the choroid. So you got pigmented layer from rods and cones. Then you have a synaptic layer between rods and cones and bipolar cells. Then you have a synapse layer between bipolar and ganglion cells. So we're going to look at a picture real quick that shows that. I think it's in the next image. If it'll click over there. Okay. This is an actual image. It's been colorized. But at the very top, you have the sclera. That would be the outer white of the eye, the collagen-filled white of the eye. The choroid layer is the pigmented layer of the pigmented separate layer, not part of the retina. The pigmented layer of the retina is right here. Here's your photoreceptors, rods and cones right here. Then you have where they synapse with the bipolar cells, and what you're seeing are cell bodies. All those dots are cell bodies. And then here's where you these cells synapse with the ganglion cells, and then here's where the axons of the optic nerve begin. So the light would be going in that direction. Now I have another picture that I think is a little bit, a little clearer or easier to see, but it's upside down from the previous one. The sclera is at the bottom, then the choroid, then the pigmented layer. So notice the direction of light is coming from above this time, as if you're looking up in from this angle. You're looking up and the light's coming into your eye and passing through all these layers until it hits, that light shown in yellow hits the rods and cones. So here's the cones and rods. Cones are important for color vision, rods for dim light, like nighttime vision. So there you've got the bipolar cells and the ganglion cells. These cells are also communicating with those other interneurons that we're not going to talk about, but there is, you can see synapses with these other amacrine cells and so forth that we're not going to worry about, but they're shown there too. So notice light moving this way, direction of impulses moving in the opposite direction. Okay, so we're going to look at how light stimulates. Oops, I don't know why there's such a delay there. Okay, as I said, the rods and cones are named for the shape of the outer segment. And here is an enlarged picture. At the top of the page is the pigmented layer of the retina, which is in contact with the choroid layer, and then the sclera would be even further up. Here are the cones, the color vision, and cone color. Cones, this is what they call the outer segment that's cone-shaped. The rods have a rod-shaped outer segment. The outer segments have folds of plasma membrane that contain the photopigments, the photopigment proteins that we're going to be looking at. So imagine that the plasma membrane forms pleats. Instead of being a flat plasma membrane, it pleats back and forth. Those are called folds. And embedded in those plasma membrane pleats or folds are the photopigments for color vision. In the rods, you have discs, which are, it's kind of hard to describe, but they're kind of like extensions of the plasma membrane. They're disc-shaped. I'll just say that. It's the easiest way to describe them. And your book talks about them. You can read about them if you want. But discs also are embedded with photopigment, but photopigment for dim light vision, dim light conditions. They're only stimulated under low light. They're not stimulated under bright light conditions. That's the outer segment. The rest of this is the inner segment, and from here down, that includes the cell body of the cell. It includes the photoreceptor cells. You can see there's mitochondria, Golgi complex, nucleus. Basically the organelles of the cell are in that inner segment. The impulse, remember, the light's traveling this way, stimulating the photoreceptor pigments in these folds, and then these cells are going to generate receptor potentials because they're receptor cells. They're going to generate receptor potentials in response to light and move that, and they're going to generate impulses that travel down, become action potentials, and or become, I should say not action potentials, but receptor potentials that stimulate the release of neurotransmitter, and from there they're going to stimulate bipolar cells. Yeah.
So whenever you light comes into your eye, does the nerve impulses come back forward and then go along the front surface?
Right.
That's where I'm kind of getting lost.
I know. Yeah, it's the opposite of what you would expect.
Okay,
You'd think
so
it would go deeper. You think the nerve impulses would go deeper, not yeah. So it's like the light is penetrating. These layers are very thin. So, and probably, the light can penetrate through there. So the light penetrates through those layers, hits the deepest layer, the neural layer, and then the impulses travel back to the surface because those axons are at the surface, and then that goes back to the optic nerve.
Gotcha.
Seems weird, isn't it? But yeah, that's how it works. And maybe there's maybe there's a reason for that, and maybe that way the light intensity isn't quite as high because it's not right at the surface. So maybe it prevents overstimulation, I would assume, from that. Now notice that in the inner segment at the very tips, this would be the side synapsing with the bipolar cells and that synaptic layer. This is where you'd have the release of neurotransmitter that then stimulates the bipolar cells. Now we're going to look at, let me see if there's something I'm forgetting about. Okay, so 120 million rods. You have a lot of rods in the retina. And this is not just we're not just talking about the fovea centralis, but the entire retina has 120 million rods, which are stimulated by dim light, allowing you to see black, white, and gray. Notice that when you're in the dark, you don't see color unless you're shining a flashlight or you have a source of light that would stimulate your cones. So the rods only allow you to see black, white, or gray, which would be under dim light or low light conditions. You have 6 million cones, and they provide color vision. They're stimulated by bright light. And you have 3 types of cones. Some of these are some of the cones are what we call red cones. Some are green, and some are blue, depending on the photopigment that they contain in those pleats, in those folds, that we were talking about. In other words, in the folds here, you have either pigment that responds to, well, it's called red, but it actually responds more than that. But what they call red photopigment, green photopigment, or blue photopigment. Okay. All right. If there's anything else I forgot here. Okay, just said all that. Let's move on. Okay, so photopigment is actually a glycoprotein. It contains, the glycoprotein is called opsin, is the name of the protein. And the glycoprotein is just a protein with some carbohydrates added to it. And there are 3 types of these glycoproteins, these opsins, 3 types in the cones, and 1 type in the rods is called rhodopsin. And the variation in those cone pigments allows for different kinds of absorption of light, as we'll see. There's also in this photopigment, there's also retinol. Retinol is a vitamin A derivative. It's made in other words, from vitamin A, which is also called beta carotene. And so you get this from eating leafy green vegetables and eating anything orange, like carrots and pumpkin and things like that are orange. You may think, well, retinol, isn't that the same stuff they put in lotions that help treat wrinkles and acne? And there's retin A, which is an even stronger version. Those are derivatives of vitamin A. In fact, retinol is the same retinol that you find in lotions that they use for over-the-counter wrinkle creams. Retin A is a little bit different, but it's also a vitamin A derivative. So retinol isn't just in your retina; it's also used as a wrinkle cream. But anyway, the absorption of light by the photopigments in the rods and cones causes structural changes in those photopigments that leads to the generation of what we call receptor potentials. Those structural changes involve what they call bleaching of the pigment, and literally the pigment turns from colored pigment to clear pigment in that bleaching. When light hits the pigment, it turns it from a colored pigment to an uncolored pigment. So we're going to look at what happens in those pigments in the rods, and well, really we're going to look at what happens in the rods, but it's a similar process in the cones. So here is a picture of, you may have to click on this twice in your notes because it looks like it kind of had, if you click on it once, you only get that. So you may need to click on it twice on your computer. But anyway, this is the bleaching and regeneration of rhodopsin pigment, which is the one for dark light conditions, dim light conditions that give you black and white and gray vision at night. So this would be happening in those discs in the outer segment of the rods. And notice that rhodopsin is in the rods. That's one way of remembering it. Rhodopsin pigment is in the rods. Here is the rhodopsin pigment. This is the opsin glycoprotein, and here is the retinol, the vitamin A derivative. And there are 2 forms of retinol. There's cis-retinol, which is a bent molecule. We're not gonna look at the structure, but it's a bent molecule. And there's trans-retinol, which is a straight molecule. So you start out with it in its bent cis form, and when light hits the retinol, it causes a change in the shape of the retinol pigment. That's called isomerization. I'm not going to worry about that term. That just means you change the shape of the molecule. Isomers are different forms of the same molecule. So you're changing it from cis to trans. Notice it's happening in response to light, specifically low light. It doesn't happen, I'll explain why it doesn't, well, it does happen in bright light, but I'll explain why you don't use these for bright light in just a moment. But so you go from cis to trans with the retinol, and this causes the transretinol then separates from the protein. It causes that, to separate. It no longer fits with the protein. That's the bleaching part. You go from having a pink rhodopsin pigment, have a pinkish color, to a colorless pigment. That's called bleaching. This bleaching is what causes the cell to generate receptor potentials. We're not going to get into all the steps of that. It's a bit more than I want to get into. But that bleaching is what causes the rods to generate receptor potentials. And you have a similar process in the cones. They are going to respond to bright light, and they're going to respond to certain wavelengths of light a little differently. But you're still going to get that bleaching. It's just a different opsin protein that you find in the cones. 3 different opsin proteins. But anyway, so you got a colorless pigment. Well, then this is going to get converted back to the functional photopigment. This is called the regeneration of the pigment. And this happens under the effect of an isomerase enzyme, that retinol isomerase, which, and don't worry about the enzyme name, but there's an enzyme that converts the opsin back to its previous form by converting trans to cis retinol, and then the cis retinol can bind back to the opsin. So opsin binds only to cis retinol, it doesn't bind to trans. That's why the trans gets released. Now, once you turn cis retinol or trans retinol back to cis, it can rebind to opsin, and you're back to where you started. Okay, so every time these pigments absorb light, they get bleached, then they regenerate, then they, more light hits them, they get bleached, and then they regenerate. Every time they get bleached, that generates a receptor potential, which then stimulates the release of neurotransmitter, which then stimulates the bipolar cells, and you send impulses down the line. Now notice, I also mentioned over here that vitamin A is stored in your pigmented layer, the retina, and that provides more retinol, so that if some of it breaks down, you have a source of it. This is why vitamin A is so important for night vision, and sometimes people have what's called night blindness. They don't have enough not enough vitamin A in their diet, and not enough vitamin A in the pigmented layer, and so they're not providing enough retinol to their photopigments. So that's why they say, eating like spinach and carrots and things like that are good for your night vision, because they contain, like carrots contain a lot of beta carotene or vitamin A, and so do leafy green vegetables like spinach. Okay. Now, when cones are stimulated by bright light, they regenerate their pigments or photopigments. In other words, they convert their transretinol back to cis-retinol more rapidly than the rods do. Rods have a slower regeneration. If you go out in bright light, your cones are stimulated, but they're able to regenerate their photopigments more rapidly. Whereas rods, if you hit them with bright light, they get bleached out, and as long as there's bright light, they stay bleached. They don't work under bright light conditions. So rods are not working during daylight out, or during high light conditions, but cones are. So cones don't work at night. That's why you don't see color vision unless you have a flashlight or something that's reflecting light to your eyes. And meanwhile, rods don't work during the daytime because they get bleached out, and they have to regenerate under low light conditions. So they're only active in low light conditions. Now, so this leads to something called light and dark adaptation. Now we talked about adaptation in neurons. This is a little bit different kind of adaptation. This doesn't mean necessarily loss of sensitivity. It's a different kind of adaptation. We're talking about in terms of vision. Dark adaptation is when you move from light to dark, like when you go from the outside into a movie theater. At first it's really dark and you can't see anything and you might trip over things. You need a flashlight to see. But after a while you notice that, oh, I can see now, I can see in the dark. It takes some minutes though for that to happen. So what is happening is when you go from bright light and all your rhodopsin pigment is bleached and you go into dark light, then your rhodopsin can regenerate and you can start to slowly get your nighttime vision back. But it takes several minutes. Those pigments regenerate slowly. So it takes several minutes. So dark adaptation is very slow compared to light adaptation. So then you go out of the dark theater out to the bright light, and at first it hurts your eyes. All that light coming in your eyes initially is almost painful. But then you adjust to that light. And that's because your cones will, when it's suddenly exposed to bright light, they will lose some sensitivity. They will adapt in terms of loss of sensitivity. They adapt in the sense that other things adapt. They become less sensitive so that you're not being deluged with all this bright light that hurts your eyes. So, but keep in mind that in light adaptation, your rods are going to get bleached and they're not going to work anymore. So it's just your cones working at that time. So eventually, fairly rapidly, your light adaptation happens pretty quickly, and that means you have a decrease in sensitivity of your cones. So how do those cones work? Whenever you're looking at a pigment, whatever the pigment is, when you're shining light on a pigment, like sunlight or light from the lights around us, that light contains all the wavelengths. Or at least sunlight does, but even indoor lighting contains all the wavelengths, all the colors of the rainbow are in white light. And remember, when you pass that white light through a prism, it divides all the colors or all the wavelengths up into the different colors of the rainbow. But, so you've got green, red, blue, yellow, all the wavelengths are there. But then when they hit something that's blue, let's say, a blue object with blue pigment in it, some of those wavelengths get absorbed by the blue and others get reflected. The ones that get reflected, that's what we see. We see the reflected wavelengths. So in other words, blue and green, or I'm sorry, not blue and green, but green and red and yellow and violet are being absorbed into the pigment and not reflected. Only the blue wavelengths are being reflected. Now if it's purple, it could be because you either have violet or purple pigment, or it could be that you're reflecting both blue and red, right? Because if you mix blue and red together, you get purple. So you could either have a purple pigment that's literally, it just is purple, it's a purple pigment, or you could have blue and red pigments mixed together, and that would, either way it would reflect as purple or violet. Yeah.
Is there a way to tell whether it's purple or red and blue, red and blue virtue? Or
I don't know. It's probably not really, because and there's so many variations on it. But I don't know if you could unless you knew exactly what pigments you had in that object, like clothing or something like that. You probably wouldn't know. You wouldn't be able to tell just visually, I don't think. I don't know of a way that's maybe a way to do it, but I don't know of a way to do it. Now, let's see. So that blue that's reflected is going to stimulate the cones in your retina. You have three different cones, and those three different cones are red, green, and blue because they have three different photopigments in them. And we're going to look at how that happens. So let me see here. I'm going to go here. So remember that anything you're seeing is reflected light. Now, if you're seeing white, like a white shirt, that is all, in other words, if it was white, not only would, you're shining white on the white shirt, it's going to reflect all those wavelengths back at you. That's going to look white. If it's a black shirt, all the wavelengths are absorbed by the black and nothing's reflected back. Black is a lack of color. Black, pure black pigment or pure black shirts or something like that is complete lack of reflected light. It's no color being reflected. Now, we have what's called trichromatic color vision. This doesn't mean we just see 3 colors, though. It just means that we have 3 cones, 3 types of cones with 3 different photopigments in them. And each cone, you have the blue cones, these are the color receptor cells. Blue cones, green cones, and red cones. And they absorb different wavelengths. The blue absorbs maximally in the blue part of the wavelength. Green absorbs maximally in the green part of the wavelengths, electromagnetic or visible light, and then red absorbs maximally in the red zone. Now they're named for, for the fact that they are, let me show you another picture. Here we go. I put 2 pictures together and it's not perfect, so this doesn't quite work. I'm gonna have to try to find a better picture if I can. But I was trying to show you the colors of the rainbow that correspond to these wavelengths. So on the 400 nanometer side, you have violet and then blue over here, and then it goes to green here. You can see, so here you can see the blue here, green, and then red, but it doesn't, this isn't lining up perfectly. So I'm gonna try to find a better picture. But anyway, I was trying to get 2 pictures put together and it didn't work exactly the way I wanted it to. But I don't think they have the waves quite right is I think the problem. But anyway, so let's just say that blue cones, their photopigment absorbs maximally in the blue part of the wave of the electromagnetic spectrum. Green absorbs mainly in the green part. Red is supposed to be in the red part, but as you can see, again, it's not a perfect lineup. So I'll have to see if I can fix that. So for instance, if you're looking at white light, where all of the wavelengths are hitting your eye, the white light is going to stimulate all 3 cones simultaneously, because in that white light you've got blue light, green light, and red light, right? It's all the colors of the rainbow. So all of them are going to be maximally stimulated. When that information goes to your brain interprets that as white. When you get maximal stimulation from your cones, in all 3 cones, that is interpreted by your brain as white. If you get no stimulation of your cones, that's going to be interpreted as black. So no stimulation of cones is black. But we don't just have blue, green, and red vision. We also have yellow and violet and all these variations. So notice that blue absorbs maximally there, but it also absorbs some wavelengths over here and over here. Green absorbs a fairly wide range of wavelengths, just different amounts here, because this is amount of absorbance of light. And then red absorbs other wavelengths as well, so there's some overlap in there and where they absorb. So for instance, I'm trying to see an example we could use, like yellow, though it doesn't correspond perfectly, I think blue actually comes over into the yellow over here a little bit. So if you have a little bit of blue light absorbed, and then, let's see, if this is yellow, this much green, and this much red, I'm just kind of going up like this, and I think the blue actually comes in a little bit. If you have a little bit of blue cone stimulation, a little bit of green stimulation, and a little bit of red, and a lot of red stimulation, your brain interprets that as yellow. In other words, it's getting information from these cones, either a lot of absorbance or a little bit of absorbance from those 3 cones. Your brain puts that together as a color. So it's taking the relative absorbance of these different colors, putting it together as a color experience for you, as perception of color. So if you're just seeing blue, then you're stimulating primarily the blue cones, but you may also be stimulating the green and red cones as well a little bit, and that may change the hue or the shade of blue that you're seeing. It's going to make it a different shade of blue. So the different shades of color, the different types of color, and the ability to see violet, for example, or yellow, depends on how much stimulation of these cones you're getting, and your brain puts that data together. Yeah.
So it's like if a color seems more pale or pastel to us, more of the wavelengths are absorbed than are stimulated?
Well, if it's pastel, it's probably got the pigment more spread out. It's more spread out on a white background. So that's probably what it is. And so, yeah, so if you have a strong color, there's going to be more pigment there. It's going to have a stronger stimulation, whereas a little bit of pigment is going to weakly stimulate it, and that's also going to be factored in as well. It's kind of relative amounts, relative to each other, so it's very strange, and that's how it works. And but because of this overlap, you're able to see yellow, you're able to see violet. Now, whether you're seeing violet because it's true violet pigment, it would be over here, a little bit of blue stimulation of blue cones would give you violet, but also, like I said, if you've got red and blue hitting your eyes from two different pigments, then your brain is going to say, okay, red and blue, that's purple. You can't tell whether it's a violet pigment or a red and a blue pigment. Your eye, you can't discern it, visually. So there's different ways that color can work, in other words, but it's based on relative stimulation, the wavelengths of light that are being absorbed, the amount of stimulation of each of those cones, and which cones are being stimulated. That determines color. Now, color blindness, there's different kinds of color blindness, but the one that you mostly hear about is red-green color blindness, and this is an X-linked trait. Now, I'm not sure if I don't think all cases of color blindness are X-linked. Some of them are, and some I think aren't, but there are X-linked genes. They're the genes that code for opsin, which is what the opsin that gives you color vision. Those 3 genes that code for the 3 different opsin pigments are on the X chromosome. And so, this is a recessive gene. So if you're male, you're XY, you inherit a recessive gene for one of these rods, or one of these, not rods, one of these cone pigments, you can end up with color blindness. If you're female, you have to have 2 recessive, both X chromosomes have to be recessive, and that doesn't happen very often. So women tend not, to be colorblind, but it's possible they are. Men, though, are more likely to be because they're XY. So if they inherit the allele, they have the condition. If you have a dominant allele and a recessive, you're going to still produce all of your pigments. So if you have an X-linked mutation of one of those pigments, you end up with some kind of color blindness, but I think there's some rare color blindness that's from other causes as well. And but the most common one is red-green color blindness, and that's where either the red or green cones are not producing photopigment. When it says missing, I should really rephrase that. They're just not producing the photopigment they need to be stimulated. So if you have red cones, those red cones pigments, if they're messed up, they're not going to get stimulated. Or if you have some green cones and they have a messed up protein, a photopigment protein, they're not getting stimulated. Either way, you end up with a condition where you can't differentiate between red and green. There's also night blindness. I mentioned that. That's low rhodopsin due to vitamin A deficiency. That is a different thing altogether. But you can have conditions where people have, even in bright light, can't see color if their cones are messed up. But like I said, the most common one is red-green color blindness, where you couldn't differentiate between if you saw a red apple like this one, it might look like this instead. Red and green basically look the same. And so there it's showing percentages of different types of color blindness. Normal vision is the top one, red-green color blindness. The deuteranomaly one is, you don't have to memorize these names, but that's the red-green color blindness. But you can see there's other more rare forms of color blindness, and even one where you can't see any color, it's just everything's black and white. But I can't remember if it's, what exactly is cause of some of those, because I don't think all of them are necessarily X-linked, but certainly if one or more rods have a pigment that's not working, you're going to have color vision problems. And so here's some examples of what fruits and vegetables or some tomatoes would look like with these different conditions. This is the normal vision, this is red-green color blindness, and then here's a couple of other more rare types of color blindness. They're, very unusual looking. But red-green, like I said, is the most common. But I'd have to look at the others to see if they're all X-linked. Some of them may not be. Anyway, so what it means is, if you go back to that picture back here, if one of these cones isn't working, you're still stimulating, like if you don't have red cones, you're still stimulating green cones, right? But you're not gonna get, because you're not getting any red cone stimulation, you're not gonna be able to see red. You'll see green, but you can't really see that red. If you don't have a green cone, then you're getting some red stimulation, but you're not getting that blending of those wavelengths required for seeing all those variations of green. So if you're missing one of those cones, it's going to mess up your brain's interpretation of color, and that's the issue with color blindness. Okay, now this is a picture just showing what the fovea centralis looks like in the neural layer. Notice that in the fovea centralis, which is very small, this is where that focal point is, the bipolar cells are pushed to the side, and so you're directly stimulating the cones, and you only have cones in your fovea centralis. In other words, there's no rods in the fovea centralis. So your visual acuity is best in color. You can see best in color because that's where the focal point is. These are rods over here, I guess, but these are supposed to be, I guess, cones. So that's what you're stimulating, and you're stimulating them directly. There's nothing even in the way of that, so you're getting almost direct stimulation of those cones. This also means that your rods are going to give you better peripheral vision, and this is why when you're out looking at stars and you're trying to see a shooting star, you actually oftentimes see those shooting stars shoot by like in your peripheral vision, and then you look for it and you have a hard time seeing it. It's because your peripheral vision, peripheral to the fovea centralis, your peripheral vision is better at night than your central vision. So oftentimes stars that you're looking for, or shooting stars or whatever, comets or whatever, are going to look better with your peripheral vision than with your central vision. Okay, so let's move on. Like I said, when you stimulate those photoreceptors, you're going to get receptor potentials that then trigger the release of neurotransmitter. And so let's look at what happens to that. Here's the direction that the action potentials are transmitted, but let's just look at that in these pictures. So again, these guys get stimulated by light, and they in turn they generate receptor potentials, they release neurotransmitter. The bipolar cell now generates basically EPSPs, excitatory postsynaptic potentials, running up the dendrite, and then the axon is going to generate action potentials. It's going to release neurotransmitter that in turn stimulates the ganglion cells. They're going to generate action potentials that travel down those optic nerve fibers and on into the optic nerve. Now the visual field that you see is, what they mean by visual field is what you see in one eye. And because we have our eyes at the front of our heads, we have depth perception. We're able to overlap images in our brain, which creates depth. And this is, the importance of binocular vision, by having two eyes in the front of your eyes. That way you have depth perception, so when you reach for something, you can judge the depth. And when you're walking, you can judge the depth better. Like if you notice, if you close one eye and try to walk, you oftentimes will like stumble on steps and things like that if you lose that depth perception. If you're a horse or animal with the eyes on the side of your head, you don't have that binocular vision. You don't have that depth perception. You have peripheral vision, so you can see behind you really well, and to the side where animals that might try to kill you come from, but you don't have that depth perception that we have. But primates in general, if you look at any primate, we all have our eyes at the front of our head. We have good depth perception. So how does that work? Well, first of all, this picture is showing you the two visual fields are overlapping. Where they're overlapping in blue is where you have the binocular field, which means, it means where you have your best depth perception. The light lavender areas are on the outer periphery. That's where you don't have depth perception. You have peripheral vision, but not depth perception. In the very center of the binocular field, that macular area, that's what you're focusing on. That's your center of focus. That's where you have the best visual acuity. Like I said, when you're focusing on something, everything else seems to be peripheral to that. Now, what happens in images, remember that the light gets refracted so that images get reversed. So the image coming in here is going to go over here, and it's going to be reversed here, and then the image coming in this way is going to be coming in like that. The macular, there's the fovea centralis, that's your macular region, but everything else is peripheral. So that's where the light is going, and then here's the blue lines represent, there's the fovea centralis here, where you're getting central vision, and that's that macular field, and then the peripheral field here. Light from this way is coming in this way, light here is coming in this way, and of course being the image being reversed and all that. So the light that's hitting the right side of the eye, or the image that's hitting, or the left side of the eye, I should say in this case, we're looking down on the eye. So the light that's hitting the left side of each eye, if you trace the lines, the image that goes to the left side goes to the left side of the brain. So this light image is going to the left side of the brain through the optic nerve, and this one on this left side is going to the left side as well. This one does not decussate, but this one does. Okay, so you have decussation up from the right eye field to the left brain. And then likewise, the image on the right side of the retina here is going to cross over to the, or from the left eye, I'm sorry, well no, right field. It's crossing over to the right side of the brain. The eye image here on this right side of the eye does not cross over, it just stays on the right side. So everything, in other words, everything on the left side goes to the left side of the brain, and if it's coming from the right eye, it's going to cross over. Everything on the right side is going to go to the right side of the brain, so from the left eye, it's going to cross over. That's a partial decussation. It's not like all of the fibers are decussating, but some of them are. So notice this is the optic nerve coming from the back of the eye. It goes to the optic chiasma, which is where some of those fibers decussate, and then the optic tract carries that information to the thalamus, and then from the thalamus it goes to the primary visual area. So here's another picture. This picture I think is confusing a little bit to me, but it's okay, I guess. I'm not sure I guess they're showing the same thing, but they don't show the macular field in this one. I guess this is supposed to be the zone of overlap. This is the biocular field, and this is the peripheral image. So again, you can see that light going over here to the left side goes to the left side of the brain. This image goes to the left side of the brain but decussates. Right side of the retina images from there is decussating. This one's not. That's the optic chiasm. So you have some crossing over of fibers. And because of that crossing over, that's what gives you biocular vision and gives you, or binocular vision and gives you depth perception. Yeah.
I was just going to ask, and it's really almost all the body, why is there always that crisscross where the left controls the right and yada yada?
I don't know if there's I'm not sure.
Is that just how it evolved?
Reason.
Like just
Things sometimes just evolve that way, but it seems like there'd be a benefit to that. But why that happened, if it just was a fluke and that fluke worked and that's what was retained, or if it might have had some kind of benefit, but I don't know what that benefit would be. Maybe there's something out there about
if
that.
it ain't broke, don't fix it.
Exactly. Well, that's how evolution works. If it's not, if it works, it works. If it doesn't, it doesn't. Then you might not survive if it doesn't.
I don't know.
Now this is showing
it's your whole
this is
body,
where
though.
those images go to the primary visual
Like
area of the
everything in
cerebral
your-
cortex.
no,
I'll
it's
show you that in a
supposed to do
sec.
right,
But let me show you these
vice
optic
versa.
nerves, optic chiasm, and optic
I
tract.
want to think I
real
read
quick.
something about it a
This
long
is
time
an
ago, but
inferior
obviously
view
I
of the
forgot it.
brain. So the visual pathway is from the from the back of the eye to the optic nerves. There's a foramen, by the way, at the back of your eye orbit called the optic foramen. You may remember that. It's a little hole at the back of your eye. And that's where the optic nerve exits the eye socket and goes into the brain. They cut it off, so the optic nerve is much longer than what they're showing. There's the optic chiasm, so it makes an X. There's the optic chiasm, there's the optic tract. They dissected the brain to show, because at this point it goes into the brain. It's just a short little tract. Notice it's called a nerve here and a tract here. So the name, it's kind of like the olfactory tracts. This is considered to be a tract. These are the nerves. But anyway, the tracts then run into the brain and go to the thalamus, which is, I guess, right here is the thalamus region. And then from there, you've got these, you can see all these array of fibers that then go to the primary visual area at the back of the brain, which is in the occipital lobe. So I've got a picture, I think. Well, here's the optic nerve. The pituitary gland kind of sits right behind the optic chiasm, so you can't see it from this angle. But the optic chiasm is right here, where the pituitary gland is. And then there's the optic tract, and now it goes internal into the brain. And there's where that information goes to the primary visual area, which is area 17, and the surrounding visual association area in the occipital lobe. This processes the information, and then the association area stores that as memory and retrieves memory, so you recognize what you see, so you know what you're seeing. Okay. So that chiasm again helps with creating an image in your brain of depth, because you're putting two images together inside your brain there. Okay, now there are other parts of the brain, not just the primary visual area, but other parts of the brain also receive information, visual information. Hypothalamus, there's some axons that carry the information to the hypothalamus from the eye. This allows you to create or to control your circadian rhythms, your day and night cycles. Daytime, you're getting a lot of light coming in and going to your hypothalamus, and nighttime you're not. So your brain is able to kind of control your day and night cycles and your sleeping pattern. This is why people who are blind sometimes have trouble controlling their circadian rhythm and their time of day when they sleep and things like that, because they're not getting that visual information that helps them control that. There's also, I forgot to mention this, the pineal gland, which is a gland in your brain that produces melatonin. It also it responds to high light conditions where it suppresses melatonin. Under low conditions, low light, it makes more melatonin, which is what makes you sleepy. So hypothalamus and pineal gland are involved in that. Cerebellum also is monitoring visual information. Remember, the cerebellum is receiving signals not only from your eyes, so you're just visually seeing your body position, but also from proprioceptors and equilibrium receptors that we'll be talking about, the vestibular apparatus or equilibrium receptors, and putting all that information together to help you maintain your posture and balance. Okay, so that's it for the eye. Any questions about that before we move on to the ears? Okay, any questions? Okay, so here, the organs of hearing. We're also going to look at the organs of equilibrium. All of these are housed deep inside the lateral side of your skull in a structure that looks like a snail with a weird head. This part is called the cochlea. And cochlea is named for the snail. I think it has something to do with snail in Latin. And it does, it looks like a snail shell. Your organs of hearing are here in the cochlea, and your organs of equilibrium are here in what's called the vestibular apparatus, which is kind of the head of the snail. There is a bony space. You can see how the bone fits around that space there. We're going to look at that in just a minute. But that's called the bony labyrinth. The bony labyrinth is the bony space, a tiny space that fits the shape of this snail. So the bone, if you were to cut open the bone and look deep inside, it's a small structure, but you'd be able to see the shape of the snail kind of that conforms with that. There is fluid here. It's called perilymph. There's fluid that surrounds the cochlea and surrounds the semicircular canal. So, fluid in that space there. Now, going to the outside, this is the ear canal. We're going to come back to that in a second. Here's the outer ear. The outer ear is called the auricle, and you have different parts of the auricle, the helix and the lobule or earlobes. The helix part is cartilage. It's elastic cartilage versus the lobule, which is, areolar connective tissue and fat inside there under the skin. So if you pierce your ear here, it's very vascular and doesn't get infected as much as here, where you've got cartilage, which is avascular and tends to form scar tissue if you're not careful, and infections if you have cartilage piercings. Anyway, together this is called the auricle or pinna, and the auricle kind of gathers sound waves. Sound waves coming to your ear are kind of funneled by the auricle into your external auditory meatus, which is your external ear canal, the ear canal here. And that carries sound waves to your eardrum, which is called the tympanic membrane. It's a membrane that extends across the external auditory meatus or ear canal. And that is your outer ear. And then you have the middle ear region here. The middle ear is connected by the auditory tube to your throat, the nasopharynx. I'll show you where that is. That's part of your throat behind your nose. And this helps equalize air pressure between the outside, because your nose is connected to the outside, and that pressure can change, as you go up and down in altitude, and as weather, if you have high and low pressure systems, that can put pressure on the eardrum. So having this connection to the outside, basically through your throat, that equalizes air pressure, which is why when you chew gum and swallow or yawn, you're forcing air up the auditory tube, and that helps, it makes that popping sound, but that's equalizing the pressure, so that takes off the pressure from the eardrum, which could, if you didn't do that, could rupture the eardrum. This is also called the eustachian tube. You've probably heard it called eustachian tube, but the other term for it is auditory tube. That's the middle ear, and this is often where children get ear infections. They get a buildup of fluid because in children, their auditory tube is more parallel and not slanted. And because their auditory tube is more parallel, it doesn't drain when they have inflammation from allergies. And then that fluid gets bacteria in it, the bacteria grow and cause more inflammation, and that causes the eardrum to bulge, can cause the eardrum to rupture. And then the eardrum does heal, but it can cause scar tissue to form. So you don't want that to happen too often. But as children grow, the auditory tube becomes more slanted and can drain better and more efficiently. So usually those ear infections go away as a child gets older. Now in the middle ear you have three little bones, the malleus, incus, and stapes. They transmit vibrations to a little structure called the oval window, which is where the stapes comes into contact with this part of the cochlea right here. And that transmits vibrations to the organs of hearing. So you're responding to the vibrations of sound waves. The sound waves cause vibrations here, which are transmitted to the ear, or to the organ of hearing. So let's look at some of these things. First of all, sound waves. There's different aspects of sound waves which can be measured because they are waves. They can be measured in terms of frequency, which is waves per second, how many waves are arriving per second. And that is what we interpret as the pitch, high or low pitch. The higher the frequency, which means the shorter the wavelengths, the higher the pitch. And this is measured in hertz, and this just gives you some ranges, just so you know what we're talking about. 20 to 20,000 hertz is the audible range of frequencies that we can hear in terms of pitch. Speech sounds fall generally in this range, from low pitch, deep voice, to high pitch. And so here's what we're talking about. If you're hearing thunder, that would be a low frequency or long wavelength sound wave, dark, deep booming sounds. And then if you heard a mouse squeaking or a very high pitched sound, that would have shorter wavelengths and higher frequency in terms of waves hitting your eardrum per second. Okay, so frequency, you can look at this either as wavelengths, or you can look at it as frequency. Hertz is frequency, in waves per second. Then you have, where are we? Okay, I've got about 3 minutes, so give me a sec, give me 3 minutes. Sound intensity or loudness is measured in decibels, and decibels is a logarithmic scale. It's like pH, it's a base 10 logarithmic scale. Each decibel is 10 times higher or lower than the next one. So 1 would be 10 times more than 2, you know. So, or I should say 2 is 10 times more than 1, let's say. So the hearing threshold is considered to be 0 decibels. So the sound that you can barely hear as a young adult would be considered to be 0 decibels, and then it goes up from there. So this gives you some examples. Anything really loud, though, can be painful, can elicit pain responses. Yeah.
Do children hear better or worse?
She takes math classes.
They probably hear better, partly because they tend to hear like pitch sounds change as you get older. We'll talk about why that is later, but they probably have a little bit more sensitive hearing, partly because the cells that respond to sound are little hair cells. They have little hairs that vibrate, and those hair cells can get damaged by a lot of sound. So yeah, as you get older, your sound ability to perceive sound gets a little less. Now, wait a minute, it's 10:44, so I've got a minute. So I just pointed out some of these structures already. The external outer ear I talked about, the auricle is actually also called the pinna. And by the way, there's ceruminous glands. They produce the earwax that catches debris and keeps out bugs and things like that. Your middle ear is where you have the auditory ossicles, which are those little bitty eardrum or ear bones. Malleus, incus, and stapes. This is also where the auditory tube or eustachian tube is. And then the inner ear is called the labyrinth. You have that bony labyrinth that structure fits into, and there's fluid-filled space called perilymph in there. What we're going to do is talk about hearing equilibrium, I guess, on Tuesday. If you can finish that up.